For lab number 7, we get into the topic of Mendelian genetics. Now, the first thing I want to say is that if you have not watched the lecture videos yet, please go watch the lecture videos first, because I believe it will answer a lot of questions that come up early in this lab. So if you haven't done that, go ahead and pause now, come back and watch this when you're ready to do the lab. But let's get started. So genetics is the study of inheritance. In other words, it's the study of how traits or characteristics of an individual are passed on. So we have a couple of terms we need to start off with. One is a gene. So a gene is a specific sequence within our DNA that codes a particular trait. So this is going to be something like eye color, or a gene involved in height. Maybe it is a feather type or fur type. These are something that codes a particular trait about an individual. Now, for our purposes, traits come in two varieties, or a few varieties. These are alleles. So alleles are versions of genes. So for our eye color gene, we might have a blue, a brown, a hazel, a green allele. Or we might have just two. Let's say we have fur color and we have straight fur or curly fur, or long fur or short fur. These are alleles of particular gene types. Now, there are different ways that alleles interact. For this lab, we are going to look at dominant versus recessive alleles. Now, our lecture goes into far more detail here, but for our purposes today, we are going to say that the dominant allele covers up the recessive in the heterozygous genotype. The recessive allele is the one that is covered up by the other, and also requires two copies of that allele to show the phenotype. In other words, we only see it in a homozygous genotype. On the other hand, that dominant allele can be seen in the heterozygous genotype or in a homozygous genotype. Now, just a quick review of these terms. Remember that a genotype is the genetic makeup of an individual. Now, we're going to note our genotype using letters. We're going to use a dominant an uppercase letter for a dominant allele, and a lowercase letter for a recessive allele. Now, as we learned in previous portions of our course, genotype directly affects our phenotype, or essentially the physical appearance or a physical characteristic that we see of an individual. So if our trait that we're looking at is, let's say, corn seed color, and we have two versions, yellow or black, our phenotype is going to be yellow or black, and each of those phenotypes is going to be related to the genotype and the particular genotype combination or allele combination that they have. Now, one of the first things we're going to ask you to do in this lab is to determine if a trait is dominant or recessive. Now, you determine this by looking at the alleles, but you can also determine that by looking at the offspring for some information. Now remember, there's a ton more information in our lecture, so you might need to supplement with that. But our recessive is often covered up in a generation, even if both parents, even if one of the parents has the gene. So let's say we have a corn that's yellow and we cross it with a corn that's black or purple. Some people call the color purple, black or purple. Now, if we make this cross and each of these individuals are homozygous, we make this cross and we look at our offspring and all of the offspring are yellow, we know that yellow is dominant because it covered up the black. We also know something about the genotype. The parent that was yellow could only pass on a dominant allele, and the parent that was black can only pass on a recessive allele. So that means that this individual is heterozygous. And again, if we look back at our definition of recessive, we see that the recessive allele is covered up in the heterozygous. There are no black or purple seeds because they're being covered up. For this week's lab, we are going to be looking at two different types of characteristics in corn plants. We are going to be looking at seed color. These colors are going to be yellow and black or purple. Sometimes the manual says purple. And seed shape. And their shapes are going to be round or wrinkled. Now, sometimes students get the wrinkled mixed up with the dried. Sometimes when the kernels are dried, they get sort of like one line down the center where that sort of looks like it might be wrinkled. But the wrinkled is actually kind of shriveled and very, very dry looking. So those are the two characteristics that we're going to be looking at today. And we are going to be using the concept of a Punnett square to determine the possible offspring that two parents can produce. So the first thing that we want to do is determine parental gametes, parental genotypes. So mom is going to have one phenotype and dad is going to have, a genotype. Mom is going to have one genotype, dad is going to have one genotype. And those are going to be obviously related to their phenotype. In this case, one parent is going to be yellow colored, the other is black or round and wrinkled. So let's say that mom is heterozygous and dad is heterozygous. Now we know this either because we have tested or we have looked at their parents or we've been told. Now, if mom goes through meiosis to produce eggs, her eggs, because of independent assortment, law of segregation, because of the law of segregation, her gametes, her alleles are going to split into separate gametes. So her gametes are either going to have the A allele or the little a allele. Now those are the only two possibilities that she can pass on, and she only passes on one to each child. Same with dad. He passes on one A or a little a. So this is basically the result of meiosis and possible gametes. Now, to determine the possible outcomes of fertilization between mom and dad, we're going to use a Punnett square. So let's say that mom contributes big A or little a. We don't know. and dad contributes big A or little a. Now these are their gametes, so these are their possible gametes. And inside this Punnett square, we're going to see the possible fertilization events. So when this happens, one possibility is the homozygous dominant. You can also have homozygous recessive. There's 2 chances of that, and 1 homozygous recessive. Now from here, we are going to get a particular phenotype ratio. So these 3 are all going to be the dominant genotype. Now we'll ask you in the lab to determine which genotype that is. And you do that based on the parents' given genotypes and the phenotypes that you see in successive generations. And then 1 is going to be the recessive phenotype. So we would expect an offspring ratio of 3 dominant to 1 recessive. Now what this means is that if you have a large number, or even just a small number, but a large number in a population, you can assume that roughly 75% of your individuals are going to have the dominant genotype and 25% are going to have the recessive. Now, it's never going to come out perfectly just due to chance. It's like if you flip a coin 100 times, there's a good chance you're going to get 50 and 50, but still within reason, you might get 48 and 52 heads versus tails. So we would expect that we would have about 75% dominant and 25% recessive. Notice that if you do a cross between 2 heterozygous individuals, you will always get that 3-to-1 ratio. That ratio is important. So if we have 100 offspring, we expect 75 dominant and 25 recessive. If we have 500 offspring, we would expect 375 dominant and 125 recessive. So no matter how many we have, we would expect a quarter to be recessive. If we only have 2 offspring, so we go back up here, if we only have 2 offspring, there's still a 75% chance that each of those offspring would be dominant. So we can use this information a little bit in slightly different ways. So in this lab, we are going to ask you how many you would expect of a total number. So you're going to count your corn, and let's say you find that you have 212 kernels. You would expect 75% of those to be one color and 25% to be the other, and you will have to calculate what you actually expect. And then we are going to ask you, does that make sense? Do you have roughly that 3-to-1 ratio? So let's talk about each section really quickly before you get started. In 7.1, we are going to ask you to determine which trait is dominant and which is recessive. And we're going to tell you that the parents are true breeding. So true breeding means that they always produce the same type of offspring. And the reason that they do that is because they are homozygous, either homozygous dominant or homozygous recessive. And we're going to give you some information about the parents. We're going to give you a parent that is yellow and a parent that is black. And we're going to say the first generation of offspring is a particular type of corn, either yellow or black. Now you know which of these traits is dominant and which is recessive because your F1 individuals can only be one type. If this individual is dominant and this is recessive, the yellow is dominant and black is recessive, then all of your offspring have to be heterozygous, and they would only show yellow. If, on the other hand, we go the other way, if yellow is recessive and black is dominant, when we have heterozygous individuals, these individuals are going to be black. So you can infer which of those is dominant and recessive based on the parents and the first generation of offspring. Because the first generation of offspring from this combo, big A, cross little a, can only give you one phenotype. In 7.2, we look at something called a test cross. Now, a test cross is basically a puzzle worked backwards. So we are telling you that one individual is, or shows the homozygous dominant phenotype, but we don't know if they are homozygous recessive or heterozygous. But we can figure this out by doing a test cross. So we are going to cross this unknown individual, this individual whose genotype we don't know. We only know that it has at least one dominant allele because of the color. So we're going to cross this unknown individual with a homozygous recessive. And then we're going to look at the offspring and the number of individuals, the ratio of offspring and the traits that they have. Now, I say that we work this backwards because you can actually start this problem without even looking at the offspring, and you can set it up two problems. You can say, well, what if it is homozygous dominant and I cross it with a homozygous recessive? We do our Punnett square, and all individuals are heterozygous, but they are also all dominant. So if you perform this cross where you cross your unknown individual of a particular color with the recessive and you get all one color, you know that this had to be the genotype. The other case, if we have a heterozygous individual, this one we don't know, and we cross it with a homozygous recessive, if we do that and we look at the individuals, we'll see that we would expect 50% dominant and 50% recessive. So we can look at the offspring that we produce in that F1 generation and see which pattern it follows and work backwards to determine which genotype they are. In 7.3, we're going to look at a dihybrid cross. A dihybrid means that we are crossing individuals and looking at two traits. So di means two, hybrid means cross. So we're looking at a cross with two traits. Now we are going to want to determine what chance our offspring has of having combinations of two different traits. So we're going to look at color and we're going to look at seed shape. So we're going to start out with our parents with this genotype, with the heterozygous genotype. Now, the first thing we need to remember is that one of these is mom and one of these is dad, and they are contributing two traits here. So all of their gametes need two alleles. One allele that is a Y and one allele that is an R. Now, whether that is a dominant or recessive, a capital or a lowercase, depends on the gamete. But each of your gametes needs to have two alleles, one for the seed color and one for the seed shape. And that is probably the biggest mistake that students make. You need a combination of two alleles because you're looking at the combination of traits. So to get this problem started, we need to figure out the possible gamete combinations. So for the parent on the left, the combination of Y and R is going to look like this. We can do a big Y and a big R, so there's one possible gamete. We can do a big Y and a little r. We can do a little y and a big R, or we can do a little y and a little r. So those are the possibilities for our gametes. Now the other one is exactly the same. And if you recall from lecture, I gave you a little trick for this, and that little trick is a math trick, and that math trick is FOIL, first, outer, inner, last. You use that to multiply parentheses. We're going to put the same idea here. First, big Y, big R, outer, big Y, little r, inner, little y, big R, last, little y, little r. So those are the possible gametes for the other parent. Then we're going to do our potential cross. So we're going to make a big, huge table of gametes. It's going to be a 4x4 table with the gametes from one parent and the gametes from the other. It does not matter which parent you put on the top or the side, as long as you keep all of that parent's gametes together. So all of the gametes from this individual all came down here. All the gametes from this individual came here. So as long as you don't mix them up, it's okay. And then we are going to, in the inside, show all of our fertilization events, all of the possibilities. So if we were to have 16 offspring, this is the offspring that we would expect. It doesn't mean it's always going to happen, but it should be pretty close. So we just go through big Y, big Y, big R, big R. That's one individual. That particular individual is dominant for color and dominant shape. Now you have to tell me which color. So you would say they are yellow and round, or green and wrinkled, whatever these actually correspond to. But I'm not telling you since you have to figure it out. Next one, big Y, big Y, big R, big R, big R, little r. That's another dominant color, dominant shape. Over here, we have little y, little r, little r. So that's going to be the recessive color with the recessive shape. So you fill those in, and then you're going to tally them up, and you're going to write out your ratios, your phenotype ratios for this one. You don't need to give me genotypes here, but phenotype ratios. So you need to figure out which color and seed shape each one of these individuals is. In the last part, we have a set of practice problems. So those are your simple genetics problems that you're doing for practice this week.